Good morning. It's April 25th, and this is your daily brief in science. Here's everything you need to know. A new study published in the journal Allergy has revealed a breakthrough in peanut allergy treatment, demonstrating that 18 adults were successfully desensitized. Remarkably, by the end of the trial, 2/3 of these participants were able to consume the equivalent of 5 peanuts without experiencing allergic reactions. Professor Lucy Chappell from the Department of Health and Social Care noted that these findings represent a significant milestone in allergy treatment, potentially improving outcomes for patients. However, it's important to highlight that despite these successes in desensitization, participants still retain their allergies and must avoid exceeding their daily dose while keeping an EpiPen on hand for emergencies. The research was conducted by King's College London and Guy's and St Thomas's NHS Foundation Trust, emphasizing that this therapy requires close medical supervision. Experts have cautioned against self-treatment, particularly with oral immunotherapy, which should not be attempted independently. While the study's results are promising, experts are also aware of its limitations, including a small sample size and a short follow-up period, which necessitate further research. Out of the initial 21 participants, 3 dropped out due to allergic reactions and 3 for unrelated reasons, leaving 15 to complete the study. Blood tests conducted during the trial indicated increased levels of IgG antibodies post-treatment, which may help counteract the allergy-causing IgE antibodies. Participants like Chris Lassiter, who has lived with severe peanut allergies since childhood, expressed the anxiety and vigilance required to avoid allergens, especially after experiencing life-threatening reactions. This study is particularly noteworthy as previous desensitization treatments primarily focused on children, leaving adults without similar options until now. The trial duration varied, with some participants experiencing delays due to the COVID-19 pandemic, extending the process to 23 months for some individuals. Chief Investigator Stephen Till emphasized the need for further research to confirm these results, reiterating that such treatments should only be conducted under professional oversight. A remarkable discovery has emerged from northeastern Brazil, where a newly identified fossilized hell ant named Vulcanidris cretensis may be the oldest ant ever found, dating back approximately 113 million years to the Cretaceous period. This significant find was made by Anderson Lepeco, a researcher at the University of São Paulo, while examining a collection from the Crato Formation, known for its exceptional fossil preservation. The research team, supported by funding from FAPESP, published their findings in Current Biology under the title, "A Hell Ant from the Lower Cretaceous of Brazil." Using computed tomography, they confirmed the fossil's classification as a hell ant, revealing its distinct headgear and specialized scythe-like jaws utilized for predation. Microcomputed tomography imaging suggests that Vulcanidris cretensis is closely related to hell ants found in Burmese amber, indicating a potential global distribution during the Cretaceous. This discovery aligns with genomic studies proposing that ants first evolved on the supercontinent Gondwana before spreading worldwide. Phil Barden, an evolutionary insect expert, noted that this fossil extends the ant record by approximately 10 million years, offering valuable insights into ant evolution. The fossil also indicates that hell ants thrived in diverse habitats, showcasing their adaptability to both dry environments like Brazil and more humid tropical locales such as Myanmar. Hell ants are recognized as a transitional lineage, exhibiting wasp-like characteristics, including complex wing structures that reflect their common ancestry with wasps. Lepeco emphasized the importance of examining existing collections for paleontological discoveries, highlighting Brazil's underexplored fossil insect fauna. The specialized feeding apparatus of this ancient ant suggests that even early ants had developed complex adaptations, markedly different from those of modern ants. New research from the University of Georgia reveals that baker's yeast can trace human migration patterns, providing insights into the historical connection between yeast and our ancestors who domesticated it around 7000 BCE. This study, led by PhD candidate Jacqueline Pena and Professor Dauda Bensasson, explores the complex interactions between wild and domestic yeast populations. By analyzing yeast DNA from various regions in America and Europe, the researchers discovered unexpected genetic similarities that suggest a history of microbial hitchhiking alongside human populations. The team utilized tree bark samples to stimulate yeast growth, which indicated that the separation between yeast groups occurred during the last ice age. Published in the journal Molecular Ecology on April 24, 2025, the study emphasizes the significant role of even the smallest organisms in narrating broader stories about human history and development. Bensasan raised concerns about how human activities may unintentionally alter microbial ecosystems, warning of the unknown consequences of such changes. The findings suggest that wild yeast populations are not completely isolated from human activity, indicating ongoing interactions over time. Interestingly, yeast from southern European winemaking regions was found to resemble strains from the southern U.S., likely due to the introduction of North American vines following the Great French Wine Blight in the 1850s. This exchange of yeast strains underscores the interconnectedness of humans and yeast in shaping environmental changes throughout history. Pena described the interaction between wild and domestic yeasts as a unique duality influenced by human actions. Bensasan emphasized the need for further exploration of yeast populations in America and Europe, building on previous findings about their differences. Overall, the research highlights the significant impact of human activity on yeast populations and raises awareness of the potential environmental consequences of these interactions. Recent research has revealed a noteworthy shift in the types of icebergs recorded in seafloor sediments, particularly transitioning from large tabular icebergs to smaller keels. This change was notably observed during the collapse of Antarctica's Larsen B ice shelf in 2002, raising critical concerns about the implications for sea level rise and climate resilience. The study emphasizes the essential role that ice shelves play in regulating the flow of ice into the ocean. Dr. Kelly Hogan pointed out that the data provides invaluable insights into the catastrophic collapse of ice shelves and the potential future changes in Antarctica as a result of climate change. Researchers from the British Antarctic Survey gathered evidence for this transition while analyzing seismic survey data intended for drilling sites in the Witchground Basin, located between Scotland and Norway. Utilizing detailed 3D seismic data, scientists identified deep grooves on the seabed, which they attribute to the keels of large tabular icebergs that broke off from the ice sheet during the last glacial period. These massive tabular icebergs, discovered near Aberdeen, Scotland, were estimated to be between 5 to several tens of kilometers wide, leaving distinctive comb-like grooves on the North Sea seabed. The transition observed in the North Sea aligns with the rapid retreat of the British and Irish ice sheet, which receded by 200 to 300 meters per year at its edges during the same timeframe. Understanding these transitions is crucial for predicting potential rapid mass loss from Antarctica under current climate conditions, as highlighted by co-author Dr. Rob Larder. The findings challenge the prevailing notion that large calving events of megabergs signal the imminent collapse of Antarctica's ice shelves, suggesting instead a more complex process of gradual disintegration. This research underscores the potential consequences of ice shelf loss, as the instability of these shelves can lead to accelerated glacier movement and increased sea level rise. Published in the journal Nature Communications, the study sheds light on how historical climate changes may inform current global warming trends affecting Antarctica. Emma Mackey from the University of Florida noted that satellite data indicates flat trends in tabular iceberg size since the mid-1970s, suggesting that large calving events are not necessarily alarming indicators of instability. A recent study has revealed concerning levels of microplastics in the arterial plaque of patients who have experienced strokes or vision loss. This finding suggests a potential link between microplastics and cardiovascular diseases, a connection that warrants further investigation. Led by Dr. Ross Clark from the University of New Mexico, the research examined 48 participants, categorizing them into 3 groups: those with healthy arteries, individuals with plaque but no symptoms, and those with symptomatic plaque. The study found that patients without symptoms had plaque containing 16 times more plastic than that found in healthy arteries, indicating significant health disparities based on the presence of arterial plaque. While the presence of nanoplastics did not cause immediate inflammation, they were associated with changes in genetic activity that may influence plaque stability and the body's inflammatory response. Additionally, genetic differences were observed in stem cells linked to the prevention of heart attacks and strokes, suggesting that microplastics could affect their functionality. The study highlights the role of micronanoplastics, tiny particles that result from industrial processes and the degradation of larger plastics. These particles can easily disperse and penetrate living tissues, raising concerns about their impact on human health. Researchers are now advocating for a shift in focus towards studying nanoplastics, which are even smaller and more pervasive than microplastics. The methodology for analyzing microplastics involved heating plaque samples to over 1,000 degrees Fahrenheit to vaporize the plastics. However, this approach faced challenges in differentiating between lipids and polyethylene. It's important to note that the study has limitations, including a small sample size and difficulties in distinguishing between lipids and plastics, which may affect the results. As the findings from medical meetings are preliminary, they require validation through peer-reviewed publication before any definitive conclusions can be drawn. The research team is planning further studies to investigate the impact of microplastics on the immune system, emphasizing the need for caution in interpreting these early results. This has been your Daily Brief in Science. To read more about these stories, follow the links in the episode bio. You can also subscribe to these updates via email at www.brief.news. For more daily podcasts about the topics you love, visit brief.news/podcasts. We'll be back Monday with everything you need to know.